3. Let's see. Hi there, very welcome to this webinar from the Testa Center, together with Jesper Hedberg from the Testa Center, Patricia Raleiras from Pall, and Suparna from Uppsala University. We're here to tell you about the filtering technology from Pall, together with the Testa Center, and also with examples together with Suparna. And you will hear first Patricia, and then Suparna, and then Jesper. So before we begin, I would like to introduce you to the platform that you're using. You see the stream in top of your screen, and in this stream is also distributed by the YouTube platform, which makes it available also after the webinar. You can also always pause this webinar and continue to watch it later. And below the webinar, you can see the Mentimeter tool. This tool is used for questions to the audience, so that you can ask your questions during the webinar to any of our speakers. So during Patricia's talk, there will be questions collected for Patricia during the talk. And then in the end of her talk, we will bring those questions up in a moderated way. So please ask questions during the webinar anytime, and we will take those up for discussion in the end of each talk. Patricia, I have read that you describe yourself as a biochemist that is fascinated by proteins, which is very interesting, I think. You've been with the Pall Biotech for 1 year at least, as I understand it, as a bioprocess specialist. And before that, you went to Medicago for 2 years as a process engineer, upstream, downstream, I guess midstream also, perhaps. You've been with the Uppsala University also, what might interest some of our viewers, at least for 6 years or so as a protein engineer, researcher. And for that, Stockholm University. So with that kind of a small introduction, I also left something like the thing that you didn't know about Patricia, which I found out she has been volunteering for the Wikimedia Foundation once in a while. So with that, I would like to leave it to you to take it away on filtration.
Thank you, Jens. It's true, it's been a while since I've done some work for Wikimedia, but it was very enriching experience when I did that. Maybe some other webinar we could talk about that. Okay, let me see if I can share my screen. So good day, everybody. Thank you for joining. I hope that you will enjoy this. My name is Patricia. I'm a bioprocess specialist working mostly on the Nordic region for Pall Biotech. In our organization, a bioprocess specialist is somebody who helps customers with choosing and testing products from the Pall portfolio in their own processes at early stage development. So that's basic description of what I do, and a lot goes around filtration technologies because it's really one of the core businesses for Pall. So today we're going to talk a little bit about filtration technologies that we use for biopharmaceuticals, from lab development to large-scale production. We're going to focus on why filtration is important in bioprocess and where you find filtration in bioprocess, an introduction to the two different main types of filtration that you find there, which is tangential flow filtration and direct flow filtration. A little bit about types of filters and composition of filters, main applications of the different types of filtration, and how you do scale up. Just a small notice that both Pall and Cytiva, and as you know, Testa Center is a Cytiva company. They are under the umbrella of the Danaher Corporation, and this means that the companies have now more of a common goal and the possibility to give to really come together and offer an end-to-end solution for customers. So we think this is really neat. But why is then filtration important in the bioprocess? Well, why is filtration important at all? You find filtration a little bit around you in everyday life, to be honest. Maybe this morning you took a cup of coffee or tea. There you use filtration. You have the leaves or the grains of coffee retained in a filter, a paper bag, while the fluid goes through and becomes your beverage. The kitchen has other examples of filters like exhaust chimneys or your colander to separate pasta from water. Vacuum cleaners have paper bags that allow collecting dust particles, but the air goes through. More modern vacuum cleaners also have HEPA filters, so fancy filters. And of course, the pandemic has come to show us that personal protection equipment is a very important type of filtration to protect ourselves. So what is filtration? So filtration can be defined like this, that it's a process where you have solid particles that are suspended, and this is important to mention that it's a suspension in, when we talk about the direct flow filtration at least, in a liquid or a gaseous fluid. And this permits that the fluid passes through, but retains solid particles. And filters are needed for several reasons. These are some of the main ones in the bioprocess, at least. We need them for optical clarity to remove particles and aggregates that can form during the process. Bioburden control also, so that you don't have an accumulation of spores, living material that can start growing in your solutions. Some steps are very important to have sterile, and therefore they will be filtration steps that will be validated as sterilizing steps. And virus reduction also, I'll talk a little bit about how to do that with the filter later on. So if we look at the generic bioprocess, so this is a bioprocess map, let's say, and you find, let me see if I can get a pointer working here. You find an upstream part, typically, where you have your bioreactor, and you have things that you add to the bioreactor. And when your bioreactor is ready, so your cells have finished producing the product, you will then go to a clarification stage. Typically, you might think of like, yes, you centrifuge the cells away, but I'm going to show you that you don't necessarily need to do that. Then you go to what we call the downstream part of the process. And the downstream part of the process is typically composed of chromatography steps and also concentration diafiltration, that is buffer exchange steps. And this takes us all the way through to formulation and filling. So where do you find filters here? Filters are found in upstream part, in the harvest, in the concentration, in the purification, in the formulation filling. They are found everywhere in the process, actually. So to put things in a bioreactor these days, and as we go more and more towards single-use systems, which are basically very fancy bags that you put in the bioreactor so that you grow cells there and you could just remove the bag and throw that away, you can't heat sterilize media. Media formulations are also becoming very complex, and so it's difficult to have them surviving heat sterilization. So you sterile filter them into the bioreactor, as well as any nutrient additives like a glucose, something like that, to make sure that you don't break sterility in the bioreactor. Also gases that you might use to aerate and whatnot. Very importantly, it's also interesting to have a mycoplasma control of whatever is going into the bioreactor. So oftentimes we use filters that are rated to remove mycoplasma. Then for a harvest, we can use depth filtration, which is a form of direct flow filtration, or we can use microfiltration, which is a form of tangential flow filtration. And oftentimes you also find sterile filtration as a final step there. Then for chromatography and also for concentration diafiltration steps, you will very often need to make sure that the buffers are clean and nice to go in the column so that they will not disrupt the packing and will not disturb separation. So you filter these components that are going in the process. But you also filter your own product in between stages. You might be used to, if you are in the lab and you are going to inject your protein or whatever product that you are working with in a column, you will filter it through a small filter and then it goes in the column. Large scale, it's the same thing. You will need to make sure that you don't have any aggregates that form naturally with time or any bioburden that goes in the columns. Then more towards the final formulation, you might want to use things like tangential flow filtration to exchange the buffer or the solution that the product is in. And perhaps one of the most important, if not the most important filtration step in the whole process is the final drug filtration. Keep in mind that when we're talking about biopharmaceuticals, patient safety is paramount. So you have to make sure that whatever is going to the vials or whatever the final packaging is sterile. And it's very often that you find even like double steps of sterile filtration at that point. So if we look at how direct flow filters work like, what do they actually remove? They remove suspended matter. So anything that is big enough to be retained by a direct filter, such as bacteria, yeasts, debris from bacteria, viruses, but also organic and inorganic particles like debris from equipment, from gaskets, from O-rings, from packaging, from your own clothes, even if it's protective clothes. Any sort of precipitates, aggregates that can form during time. So that is easily removed with the direct flow filter. Then what you also have is the possibility of separating components or proteins that are dissolved, but that it will not do with a direct flow filter. That will do with an ultrafilter instead. So what you can't remove with a direct flow filter is soluble matter. Also things like ions, small molecules, stuff like that. That is more towards the reverse osmosis part, which is not directly relevant for bioprocess filtration. So direct flow filtration works like this, that you have a flow that is perpendicular to the filter medium, and the contaminants are retained in or on the filter. And there is not a recirculation loop. This means that whatever goes through the filter, it will be filtered. It's ready to go. You don't go back to the same filter and filter more or something like that. It should be ready filtered with one passage through the filter. Tangential flow filtration is another different type of filtration. Then it's also called cross-flow filtration, and it's a method that is a separation based on sizes. So here you can actually separate components that are dissolved in solution. You have a porous membrane. And the difference with direct flow filtration is that the feed is tangential or parallel to the filter medium. Whatever is high or bigger than the retention rating of the membrane will stay upstream of the membrane in what we call the retentate, and smaller molecules, ions, salts, anything like that, pass to the permeate. Tangential flow filtration then is applied to purification, but also very much to concentration and diafiltration. So the retained substances are actually swept away from the membrane surface. They are not staying in or on the membrane, ideally not. In fact, a lot of this feed is recirculated back to the feed vessel. Talk a little bit more about that later on. So how do filters look like, in fact? Here are some examples from Paul's portfolio. So you might be very used to filters like the mini clean pack. So they are syringe filters. And they're very easy to use. You just put them on a syringe and filter a few milliliters, and that's done. But that only works for a few milliliters. So you need, if you need to go to higher volumes, you will need bigger capsules. So the mini clean pack capsules are a type of capsule that we use for process development. All of these capsules come with very different types of membranes. We also have some capsules that are called the Nova Sip, the Clean Pack Nova. They can be inline flow. They can have a T connection that allows the inlet and the outlet to be on the same side. You see an example of these types of capsules on the Allegro MVP system, which is a filtration system from Pall. And like connected here with a couple of bags that have some solutions and just goes from one side and comes out of the other. These capsules, if you would do a transversal cut on them, you would see that you have a very tightly packed membrane. In fact, even just the size of the core and the size of the packing membrane is something that has been optimized. And one of the things that has been also been optimized in filtration technology is how the membrane is folded inside these capsules. And this that is here on the image is a so-called laid over pleat construction, which really allows to pack a lot of membrane area inside a single device. Other examples of how filters can look like are cartridges, and also something that is in between, which is the encapsulated modules. Cartridges are more used for manufacturing because you do investment in housings. You need housings to install these cartridges in them. And there's also different shapes, although the AB style is a very common standard one. Here on the right-hand side, you have depth filters that are encapsulated and then can be put in a big holder, let's say the big housing, and they are encapsulated so that you also protect the operator, so they don't have to touch the filter area directly in case it's contaminated with something.
So, Patricia,
yes,
I would like to come into the presentation a little bit because there are questions coming in, and I would like to tell the audience to tell them to make sure to ask your questions for Patricia during this webinar so that we can summarize them and ask them in the end of the presentation. So don't hesitate to go to the bottom of the page, and there might be a button that says that the presenter has changed the slide. That means that you have to click there in order to get to the question form. Thank you very much.
Okay, thank you. So TFF devices, how do they look like? Well, Pall sells flat sheet cassettes. So this is one format of TFF filters that you see. You sandwich them on a holder. One type of TFF cassettes that we have is a single pass modules, which is in fact a Pall patent. I will talk very briefly about this later on. There's also other very common TFF devices, which are hollow fibers. Cytiva sells a lot of these. And there's even ceramic modules with Pall sells these. They are more for manufacturing, to be honest, but these are also, they function a little bit like hollow fibers, let's say. So we can also classify filters according to the media that they are composed of, and we can do this division that you have depth filtration media and membrane filtration media. And depth filtration media, it's a type of media where the contaminants are stopped largely inside or in the depth of the filter matrix. So they are usually or typically at least a few millimeters thick, and particles get trapped in this tortuous path, so there's a irregular shape to the particles that compose the matrix. And so the particles get retained in them, not just because of a size exclusion effect, but also because they are slowed down by this tortuous path. There might also be absorptive mechanisms. There can be different mechanisms to retain or remove contaminants from a solution, really. In membrane filtration, you have the contaminants stopped mainly at the filter surface. It's not just at the filter surface, so you do see some penetration of contaminants, but it's not really like a depth filter. If we look at micrographs of how these media look like, you probably can get a better idea of the differences. So here you have regenerated cellulose and polypropylene fibers, which can compose different types of depth filters. They can have additives like perlite, which is a volcanic glass, or diatomaceous earth, which is also called in Swedish, it's called kiselgur. Now, I don't remember if there's another good name for that. But the thing is that they create these large gaps and crevices and tortuous paths where the particles get retained. Different additions of perlite and diatomaceous earth can give different grades of filters. With membranes are a little bit more homogeneous in appearance. They can be asymmetric in the pore size. They don't need to be completely symmetric. This example is so. In this example, thickness is about 100-150 micrometers. And these can be composed of different materials also. It can be polyether sulfone, PVDF, nylon, glass fiber, polypropylene. So that's a little bit about how filters look like. So what can we do with them? So one application of direct flow filtration is depth filtration. And depth filtration is used mostly for the primary clarification after a bioreactor step. Now, depending a little bit if your product is intracellular or extracellular, you might need a lysis step in between the bioreactor and the filtration step. But in fact, with many of the products that are used as biopharmaceuticals, they are extracellular, so you don't even need the lysis. Because the cells or the cell debris, they will be retained in the depth filters and not be recoverable. So your product needs to be in solution. And you could also use them for protecting columns. In fact, this is also an interesting application when you have anything that is prone to aggregation, or if instead of depth filtration you actually needed a centrifugation step, but you still need to clarify the supernatant because very often you will have particles in suspension. Pall has a series of depth filters that's called the P series, so for pharmaceutical grade. And they are manufactured with special materials. And well, I can't really have the time to go through all the filters that we have because there's so many. But they come in different retention ratings. And I would like to call your attention to a particular one that is called the V100P. So you might remember that I said that depth filters can be made of cellulose, and they can have diatomaceous earth as an additive. These don't have that because that sort of additive typically binds viruses and viral vectors, and so we developed these filters so that it would be minimal binding of viral vectors, so it can be used for gene therapy. Pall has gone a step further and has developed double layer filters, so that combine two of the two filters that you saw on the previous graph, where you have a primary layer that is a coarse layer that will capture the larger contaminants, and a secondary layer which is then a finer filter. Now, theoretically, you can do combinations of all of these layers, but some of them are more commercially relevant. For example, the PDK11 is a filter that includes this V100P that I just talked about as a secondary layer, but has a coarser layer on the top so that it will capture cells or cell debris. And so it's developed for clarification virus feed streams. If you have bioreactors with low cell counts, filters like the PDK4, PDH5, I'm calling them PD because that's the code that they have, PD and then 2 characters. Even the PDK7, they can be good filters for that. If you have high cell counts, then you go to the PDP8, which is something with a filter that has a much larger capacity for large cells at least. In fact, it's part of what we call the StaxMax platform, developed for clarification of monoclonal antibody streams, which includes this PDP8 and the PD2, which is a finer filter that you find here. PD2 and PDD1 are both filters that can be used after the PDP8, but also after centrifugation if you use centrifuge instead of the PDP8. Other filters have shown good results after a centrifuge, like the PDH4, so sometimes I recommend that. And for applications where you need finer filters, like after TFF, if you have still some aggregates in solution, include the PDPX, PDAX. Okay, so that was about depth filtration, but now I'm going to go a little bit over to sterile bioburden filtration. And this is something that you find all over the process. So as I mentioned before, you need to make sure that whatever is going into your bioreactor is sterile. So media, additives, gases, so any fluid that is going into the bioreactor. Then very often you will need to make sure that whatever is coming out of the upstream part is in fact GMO-free. In the manufacturing context, you might have different suites. One that you manipulate GMOs which is the upstream part, and then you might move to the downstream suite where you don't manipulate GMOs. It might even be a completely different facility. So you need to have a step there that makes sure that you don't have any bugs left. Then later on in the process, you might need things like protection of chromatography columns. This is actually a very common thing to do. You pass your product or your buffers and your buffers through 0.2 or 0.45 micrometer filters. And then, of course, there is the final filtration that I mentioned before, which is very important to make sure that your final product is sterile. And so very often you might even find double filtration steps there. The final filtration should be always as close as possible to the filling point, to the vials, to whatever is the product is being filtered into. One thing that one has to then identify, because I've talked about like sterile and bioburden, so what really is the difference? The difference has to do with how the filters are validated. I don't really have time today to go into the details about how the difference in validation of bioburden filters and sterile filters is, but they can both be 0.2 micrometer filters. That is not what decides if a filter is a sterile filter or a bioburden control filter. Well, what you need to decide is if you have a strict necessity to have a sterile effluent, and therefore then you need to go to a filter that says that it's a sterilizing grade, or if you just need to know, just to protect the columns or whatever, and then your bioburden control is fine. And then again, especially for applications like final fill or addition of nutrients to the bioreactor, you might want to make sure that you don't have any mycoplasma. So you might also want that. Now, this is a bit of a dry table to look at, I think. So we're not going to go into details about it, but it's just to show the range, and I don't think all of the filters are here actually, of bioburden sterilizing and sterilizing grade with mycoplasma control that Pall offers for the biopharma industry. And in fact, our newest filters, they are composed of PVDF or polyethersulfone. These are the most common materials. yeah, and so we recommend different filters for different parts of the process. Let's say, for example, the Super EKV filters, they are standard the standard sterilizing grade filter that we recommend for most applications. But then for final filtration, we very often recommend the DFL because it's a PVDF, and PVDF membranes tend to have less leachables and extractables. So they are better for final fill applications when you don't want to have any extraneous contaminants in your product. Yeah, then another example is the Super EXECV, which is a very good filter for viscous solutions and difficult to filter solutions. So I just want to do a little parenthesis about virus filtration. Virus filtration might be not something that you think about immediately when you're doing your first sketch of how a purification process looks like, but it's very important, especially, not only, but especially if you are producing anything in mammalian cells, because you typically will need 2 orthogonal steps in the process where you can show that you do viral clearance. So this is a regulatory demand. So one of the ways that you can do this is doing a treatment of your product at low pH, if it can tolerate that. You can also use chromatography steps and validate them in the process as something that can reduce viruses. But something that is relatively easy to implement is a virus filtration, which we do typically near the end point of the process. Pall has virus filters that are 20 and 15 nanometer removal rating. 20 nanometer are the more standard ones for virus filtration. And they are typically near the end of the process because you need to have at that point a very clean product, let's say, because this is a bit difficult filtration to do. The pores are very tight, so you typically need a high pressure to do them. Yeah, so that was a bit about direct flow filtration. So now I would like to talk a little bit about applications of tangential flow filtration. And tangential flow filtration is, I don't know about you, but for me, it's something that's a bit of a mouthful. So we usually abbreviate it to TFF. And TFF, you could broadly divided in 2 main types of application, which is microfiltration and ultrafiltration difiltration. And microfiltration, you find it more as perfusion for perfusion bioreactors, or as a method to separate cells or post-lysis cell debris from your product. Then you have ultrafiltration difiltration steps more towards the mid and end of the process, both for buffer exchange and desalting between chromatography steps to concentrate, to decrease volumes. Also to have in the final formulation when you are exchanging to the final solution your product should be in. So how does tangential flow filtration work? Let's again play the video. Yes. So a typical process setup looks like this. You have a feed vessel, feed tank, where you have a pump connected, and this will pump the liquid through your TFF device. Your TFF device will then separate the small ions, molecules that can pass through the membrane into the permeate, and everything else recirculates back to the feed tank as the retentate. So with time, the solution volume in the feed tank will decrease because part of it goes to the permeate when you're doing concentration. But if you're doing a difiltration, then you just add a new solution to the feed tank, and then you continue the process until all of the small solutes that you want to separate go into the permeate. Two very important concepts in TVFF are the transmembrane pressure, or TMP, and the cross-flow flux. The TMP can be roughly, or it can be defined as the average of the inlet and the outlet pressures. So the pressure that is upstream of the membrane minus the pressure that is downstream of the membrane. Of course, when you're circulating so much liquid through a device, you will cross pressure across a membrane, and this is a driving force that will push the solutes in your solution towards the membrane. If we would just let this happen, the membrane would clog. Since we have a flux that is actually parallel to the surface, we have a cross flow. This cross flow helps to lift these molecules away from the membrane surface, and so you don't have the same sort of clogging mechanism on the membrane that you would have with direct flow filtration. So there is always a balance between TMP and cross-flow that one needs to optimize during a TFF process. A normal profile for concentration diafiltration using TFF looks like this. You typically start with a first concentration step, then you go to a diafiltration, and the point where you stop the first concentration and start the diafiltration can and should be optimized. And the diafiltration can be done in both continuous mode or discontinuous mode. In continuous mode, you add buffer to the feed vessel so that the feed vessel volume doesn't change. In discontinuous mode, you actually do cycles that are concentration and dilution. And then you typically do a final concentration so that you adjust the concentration of your product to whatever you want. So in this, you aim to have a high protein retention. Typically, you have TMPs that are relatively high. And the membranes, we recommend always to have a pore size that is 3 to 5 times smaller than the molecule targeted for retention. Typical devices that are used for concentration diafiltration are flat sheet cassettes and hollow fiber. I would just like to mention a particular type of TFF, which Paul has the patent for, which is called single pass, and this is a very neat type of TFF because in fact, you don't have a recirculation, so your retentate doesn't go back to the feed vessel. And you might think, but what's the point of that? Well, the point is that if you have molecules that are very shear sensitive, then it will be good to avoid them having this recirculation loop the whole time. So just one passage, and you have your process done. Retentate to one side, permeate to the other. And it also allows integration with continuous processing. This subject could fill another entire hour, so I will shut up for now about single pass TFF and go forward to microfiltration that you can use for harvest and clarification. And this is a little bit different way to operate TFF as compared to ultrafiltration diafiltration, because instead of letting the TMP drive the process, you control the permeate flow, which you don't do in ultrafiltration diafiltration. So in this type of filtration, you determine the critical flux, that is to say, the permeate flux where the TMP doesn't become very stable. And if you look at the right-hand side of the graph here, you see transmembrane pressures that are lower than that 1 to 5 bars that I talked about for the other type of TFF. In fact, the TMPs in this type of process are usually below 0.5 bar. And this aims to have a high protein transmission without fouling the membrane easily, which could happen with debris from cells and from whole cells also. Paul has these recommendations for the devices that we use for this type of TFF. And but you don't find only flat sheet cassettes for this. You can also use hollow fiber. Actually, it's one of the big applications of hollow fibers, I would say myself. And also those ceramic modules that I talked about earlier in manufacturing, you can use them for this type of microfiltration. So how do you scale up filtration technologies? I'm going to focus a little bit more on how you scale up direct flow filters. But what I really want to push forward is the idea that you need to choose the right technology from the start. This is paramount because your product will interact with the filter will interact with the product. You don't know how the absence of filtration in your early stage development will actually affect the process later on. So it's important to test different filters at the different stages at as early as possible. One typical example is that very often in the lab you use centrifugation to harvest, and this is a technology that is actually a little bit hard to scale up. It is scaled up industrially for certain cell types like yeast and also bacteria. But not for mammalian cells. It's really not for that. Because you get typically not very good supernatants anyway in terms of optical clarity, and it's not very consistent technology either. For that, depth filtration is really a good choice, but in some cases it's not adequate, either because the product is intracellular, it's difficult to filter lysate sometimes, so you might go for TFF devices instead and do microfiltration. And also one has to think about what are the potential scale-up volumes. Direct flow filters might be convenient, but they can't always be scaled up to very, very large volumes. What I can tell you is that at least in Pall's portfolio, all our filters are scalable. So anything that you scale at the lab bench scale, like our depth filters, for example, which come in these small capsules called the SuperCap 50. Whatever parameters you get from these test filtrations, you can scale up to any volume by just adding filtration area. So in this particular example, you have the SuperCap 50 for lab or process development, and then when you go to pilot scales or small manufacturing scales, you can have these single capsules or these big capsules in assemblies, or you can even go to the stacks capsules that then require a chassis. This is for the depth filters. Other filters can look a little bit different. I showed this slide before, but I just wanted to show again to also push forward the idea that you need to ask yourself what you want your filter to do. Do you need sterility? Do you need mycoplasma control? So this is also a thing that you need to ask. It's not just, okay, I know that I need a filter to do something. It's also, okay, how do I want the filter to perform? What are the required parameters? What is the required quality in the end? So how do we do lab scale tests in direct flow filtration? Let me load the whole slide because it's actually a very similar way of thinking. Depending on kind of filter you are working with and what step the filter is in, you might do tests and run also later on the filters in large scale at either constant flow or constant pressure. And when you have filters that don't block at all, you have the situation here. And with the constant flow, you would have you monitor the pressure upstream or the differential pressure across the filter, and with no blockage you don't see an increase in pressure. If you have a little bit of blockage, you see a slight increase in this pressure. But with a premature blockage, you will see increase in pressure already at the start of the process. If you do a test at constant pressure instead, what you see is decreases or decays in the flow rate. Now, counterintuitively enough, you actually don't want to end up in a situation where you don't have any blockage at all. You should allow a little bit of blockage, because if you see that you don't have any blockage, then you might be using too much membrane area for your process, which worsens your process economy. So this is something that to take into mind, that you don't need to have like 100% performance. It can be just good enough. So what kind of parameters do you get out of these tests? You have things like the throughput, so how much product you can filter per membrane area. What is the operating pressure, or what is the maximum allowed pressure in the process? What flow rates or flux? The flux is basically the flow rate divided by the membrane area. Naturally, what filter type and size you need for scale up. Very often we will compare different filters during the same test, and then choose the more adequate one. What is the process time, which can be very relevant if you're going for very large volumes. And then for TFF, you might need things like the volume concentration factor, what is the optimal defiltration point that I said that it's important to also optimize, and of course parameters like TMP and crossflow. So
Patricia,
all of this, yes.
we're coming up on the time. So
Yeah.
I'm just wanted to remind you to so that we can have a question to come to the closing of the presentation.
That's good, because I am going to close in 2 minutes.
Great.
Not even that. So all of this sounds very complicated, I think. But fortunately, Pall Biotech has something called the Scientific and Laboratory Services that can help all along. There are people like me, bioprocess specialists, that can help you with the process development, with choosing the right technology to in the right step in the bioprocess. And then there are my colleagues at the rest of the organization can help you with really anything, with optimization, with scale up, with technical issues, with validation services, with qualifying equipment, qualifying personnel, etc., etc., etc. However, if you're coming to Testa Center in Uppsala, you will have the local support of the bioprocess specialist who happens to live in the same city. That's me. To support you with filterability tests and any sort of advice that you need around filtration in your bioprocess at Test Center. So, and I look very much forward to work with you if you come and test your process there. And with that, I would like to thank you, and I will be open for any questions that you might have.
Hear the digital applause from the audience. Thank you very much, Patricia. There has been some questions coming in. Oh, Jesper is also applauding. So let's see. We had some questions during your talk while you were presenting the hardware filters.
Right.
Filters, yeah, and the question was: Are the membranes manually stacked on the filters, or is that automated?
So the capsules come ready-made, and the cartridges also come ready-made. So what you in manufacturing, the cartridges are really used just for manufacturing, and it's really for large volumes. It's where it makes sense for the company to invest in housings and then just change the filters. But they come ready assembled, so it's nothing that you have to put together yourself. What you might have to put together is tubing and whatnot, so that you need. Although if you are really strict about how it should be filtered, and in manufacturing you do this very often in GMP processes, you go for single-use systems, which is that you buy the filter already connected to a bag, and the whole assembly is sterilized, is gamma sterilized. So I hope that answered the question.
Yeah. So there's another question as well. How does the stacks work for deep filtration? This is probably several questions, who knows? How does it increase the area, since the sample has to pass from the top filter to the bottom? If the top filter clogs, then what?
Because the distribution is made across all the filters. It doesn't go like first through one capsule and then to the other and then to the other. It's constructed in a way that it will go through all the capsules at the same time. Ideally, if these things are well constructed, and we hope that they are well constructed enough, you don't have a very big difference in the pressure drop that exists in the different capsules, but in fact it's not that it goes through each capsule individually one after the other, but it's more of a simultaneous thing. So when you add the area, you just add more capsules and it will be still the same distribution of liquid.
So there are a couple of more questions, and we'll come back to them later in the end of the webinar. Until then, the last question I would like to ask you is, what is the best way to get in contact with you, follow your work, learn more about choosing the right filtering technology?
Oh, that's a very good question. So, of course, you can always go to our website, pall.com, for general information. And you are absolutely very welcome to contact me through my email address, which is, maybe you can write it somewhere. Maybe it was on the
it should
website.
Be on the website, or if not, it should be there soon.
To ask any questions, really, and we can discuss a little bit more. You can also I'm also on LinkedIn, so very often I share things that are interesting in the industry that can be related to filtration. In general, we also like to have this sort of initiatives where we do webinars or we do individual trainings for companies or for academia, to go a little bit more in depth about filtration. To be honest, what I covered here, I could easily talk about 4 hours about. So,
thank you very much, Patricia. I love to hear that sometime. With that, I will head over to Suparna Sanyal at the Uppsala University. I'll spotlight you for the audience here to see. And I'm happy to have you on this webinar. You're the professor at the Department of Cell and Molecular Biotechnology at Uppsala University. And as I understand it, I've been trying to understand if you've been there for 19 years soon, or if there's a 20 years anniversary coming up soon. I don't know.
20 years, I would say.
I hope that the COVID situation has calmed down, so there will be a proper anniversary for you. And I think you've been to the Testa Center once or twice, and you've been established a scalable process that we will hear about. You describe yourself as a pioneer for demonstrating the role of ribosome and RNA in protein folding and aggregation. You teach master students, for example, in the trends of molecular biology and biotechnology. And you're supervising PhDs and master students and postdocs. And one thing that you didn't know about Suparna Sanyal, which she shares together with me, is that she's following NASA on LinkedIn. I don't know if there's an astrology interest there as well. With that, I would like to give you the word, Suparna, and let you do your presentation.
Yeah. Thank you, Jens. It's a pretty interesting introduction that you have done. I would say that ribosome, the ribosome purification project, what we have run in Testa Center. So the main protagonist in the project is ribosome. And I have been working with ribosome since I was a master student, and then I did PhD, and then soon after a very short postdoc, I joined Uppsala University as assistant professor directly in 2001. So thank you for reminding, it's 20 years in the same department, in the same office, and I grew here as a professor and director of Applied Biotechnology Master Program. So, and the NASA connection is the following, is that we are now looking at evolution of ribosome and other ribosome-associated translation factors to search for how higher cells and maybe even life originated in Earth. So we recently published one such evolution paper, which is partially funded by NASA to our collaborator. So that is the connection. So anyway, that is another interesting story. But today, I would like to start by thanking Patricia for a wonderful talk, because, myself and my group members, we are the end users of this filtration technology or whatever we have done in the Testa Center. But it was wonderful to listen that the huge breadth and depth of the technology and its incredible potential to application, not only for the purification of ribosome, but for many other particles. So I start by sharing my screen. Yeah, I guess it's, you can see my screen very well. So this talk, since I'm a professor in the university and one of the end user of the technology, will be probably very different from Patricia's talk, but I still hope that the listeners or the audience will find something interesting and exciting like what we do. So the title of my talk is Large-Scale Ribosome Purification, and then thinking of the theme of the webinar today, I would like to focus on the benefit of filtration technology in this. Below, you see like these blobs. So you can see my cursor, I guess. And in the left-hand side, what you see is like, two bread buns of a hamburger with the burger missing here. That is 2 subunits of ribosome. So one is bigger, and this we call a large subunit, and one is smaller, and this we call a small subunit. And the same subunits that you see on the right, so in the back, the bluish or the purple, that is the large subunit. On top of that, the small subunit is pasted. It's just another view. So, so before we go into the presentation, I don't know, my one illustration disappeared, but doesn't matter. So what are the ribosomes, and why should we purify ribosomes? Let me just introduce you or remind you, like this is a simple bacterial cell, and this is like a cartoon of our cells or the eukaryotic cell. And these dots that you see here, which is scattered in the bacterial cell, or in the eukaryote, it is sometimes scattered, sometimes bound to endoplasmic reticulum or other membranes. These are the ribosomes. So ribosomes are important because no cells can live without ribosomes, because ribosomes are the protein synthesis factory of the cells. And as you know, we all are made up of proteins. Our muscles are proteins, enzymes, hormones, anything that you can think of, bones, everything is made up of protein. So cells must synthesize protein. And then in bacterial cell, they have like 50,000 ribosomes packed in one cell. And in eukaryotic cells, depending on the cell or the tissue or the organ, the density of the ribosome differs. So this is a cell organelle. And in bacteria, so these ribosomes are like 25 megadalton is their molecular weight. Interesting thing is that now with the COVID vaccine, I think all over the world starts to talk about mRNA vaccine, RNA and stuff like that. But the interesting thing is that in this tiny cartoon below, animation that you can see, that this golden doubled blob, that is a ribosome. And then this line that you see, this is an mRNA. So mRNA contains all the codes of a gene, which is transcribed copied in the mRNA. And what ribosome does is that it decodes that code and transforms it in the form of amino acids, and the amino acids join to form the protein, this blue thing that you can see coming out of the ribosome. And so the ribosome, with this small animation, I want to say that it's not really, it cannot stay static in one place. It must actually translocate on the mRNA. This is a huge process, a very complicated and dynamic process, but this must go on in all cells so that the proteins can be properly produced. So why ribosome purification is interesting? The first point that you see here, that the ribosomes, especially from pathogenic bacteria or even pathogenic microbes, the ribosomes are easy drug target. As I told you, that protein synthesis is central to living cells. So that means if it's some drug, if you can block specifically the ribosomes of the pathogenic microbes, then it is easy to kill that microbe without, it is easy to kill the microbe. And that's why ribosomes are very central to protein synthesis and protein folding research. So also, nowadays, actually, we are trying to build minimal cells. We are trying to build entire protein synthesis system in the test tube, and ribosome is the main ingredient there. So that's the second reason why the ribosome purification should be done. And then when we are targeting the ribosomes with some drug, since the ribosomes are highly conserved in evolution, we would not like that those drug molecules target our or the human cellular ribosomes. So that's why it is important to study like higher eukaryotic or human ribosomes in comparison with pathogenic bacterial ribosomes. And these doing in vivo or in situ in the cells is quite difficult. That's why we have engineered fully reconstituted translation system, and the heart of those translation systems, I would say, ribosomes. And that's why we need to purify ribosomes, and that's why we need to see that how we can scale it up so that this drug screening, which is absolutely essential in today's antibiotic resistance era, will be actually feasible and useful. And then, so here, I would say that if you ignore all this, first look at this box and look at the ribosome purification. I have simplified this in just 2 bullet points. That conventionally, ribosome, like all other cellular organelle, used to be purified with ultracentrifugation. So as Patricia also mentioned, that centrifugation has been actually a very good separation technique. And conventionally, this has been used for separation and purification of the ribosomes. And usually, the ribosomes are ultracentrifuged, just not in aqueous buffer, in order to keep its integrity, because it's a very complex macromolecular complex, as you see here. This purple thing is the RNA, and this cyan or the red or orange, green, these coiled stuff are protein. So you can almost think of a head, which is like the hairs are like entirely ball of RNA, and then some proteins are like some accessories stuck onto it. So in order to keep it active, when we purify, normally huge amount of sucrose solution is used in ultracentrifugation steps. So that keeps the integrity of the ribosome and keeps its activity, but at the same time, it's a very huge hazard, and it's a, I should say, fairly messy process, and takes very, very long time, lot of effort, and of course, ultracentrifuge, which is not so easily accessible to all labs. So, at one point when we were having actually some problem with our ultracentrifuge, we came up with an ingenious idea that why don't we engineer affinity tag on the ribosome? And we did that. So on the ribosome, there is this extension. It has actually one protein, which is L12. And on the ribosome, there are 4 copies of L12. 1, 2, 3, 4. So what we did is that in the chromosome of bacteria E. coli, behind the gene of L12, we fused the nucleotides, which will code for 6 histidines. So in a way, it's actually a very small engineering. So it's just like adding 18 nucleotides. But then, of course, we had a drug marker to ensure that the insertion happened. But then what happened is that, to our surprise, the resulting strain, and the resulting strain's name is JE28, named after the postdoc Josephine Edert, who actually engineered the strain. And this J28 survived pretty happily, and it offers 4 histags, like these 4 dots, as you see in the cartoon, on the ribosome. So that means this engineered strain offers you actually ribosome containing 4 histidine tags. So of course, I mean, you can understand that then we can bypass the separation of ribosomes by ultracentrifugation. Instead, one can use just histag-based affinity purification like you would do for any histag proteins these days. And I would say that with modesty, with all due respect, I would claim that engineering this histag and making J28 strain has revolutionized ribosome prep. Because then one can avoid these lengthy ultracentrifugation steps. You can get it by affinity chromatography. So here I put the comparison. So in the conventional step, we have multiple ultracentrifugations. And these ultracentrifugations require like speed, I would say that 200,000, 150,000 to 200,000 G. So depending on your rotor and its diameter, the rotations, the RPMs are different. I'm not going into that. But you need multiple ultracentrifugations, which are easily like 12 to 16 hours or even longer. So that makes the altogether one proper ribosome prep taking about seven full working days or even longer, because we are not counting the overnight spins here. In contrast, this J28 ribosome can be purified with a single affinity column in few hours, actually sometimes less than hours. In ultracentrifugation, as I mentioned, that you deal with big volume of sucrose, and in the his affinity purification, that can be largely avoided. Point I'm saying largely avoided. I will come to a point where we still may need to ultracentrifugation in this prep, but it can be largely avoided. Actually, in small scale preps, you do not need that. The conventional purification, since it has multiple steps of ultracentrifugation and so on, the yield is actually pretty low. We have noticed that we cannot actually, we get maximum maybe 40 to 50% of ribosomes extracted by the ultracentrifugation method. Whereas with the affinity purification, since it's very fast and very robust, you can get 80 to 90% of all ribosomes in the cell extracted. And then after extraction, if you compare activity, conventional purification, the activity varies between 50 to 70% activity of the ribosomes. Whereas in the histag affinity purification, we always see 70% or even higher activity. So to cut the long story short, that the conventional purification is very laborious, time and cost required, whereas the his affinity purification is very easy and fast, and it's a very low cost method. And then the second thing is that, I mean, obviously you can see here that it's an old map, but we have put up this map just to demonstrate you that this strain is so popular that anybody almost doing ribosome research all over the world requested this strain to us. And if I put the new pins, it has traveled also in South America, in Russia. So almost looking at J28 map, you can guess who in the whole world is doing ribosome research. So after achieving that, we thought, I mean, I come to the point why I said that the ultracentrifugation can be largely avoided. Because in the histag affinity purification, you know, after elution from the affinity column, you get the ribosomes in very good quantity, but still the concentration is low. And you need to concentrate it further in order to allow proper storage by shock freezing in minus 80 degrees centigrade and for future use. And in that step, we were handicapped and we had to use ultracentrifugation. And whenever we use ultracentrifugation for concentrating ribosomes, we have to remember that then we are limited in volume because, you know, the rotors have definite volume capacity. And more rounds of ultracentrifugation you add, each ultracentrifugation for concentrating can be overnight. So more days you add and more components you add, more cost for the person working also you add. So that was a bottleneck. And then we were fortunate to be able to get a project slot through Uppsala University's agreement with Jesper Hedberg's Testa Center. And we, with incredible help from them, And thanks to my brilliant group members who have actually developed the entire process almost automated, and which gives very high quality ribosome in very small time. So very little time. I will quickly go through the steps and then focus on the filtration steps. So here, first of all, we did the upstream step is that we started growing J28 in bioreactors, which we haven't done before. So that is already one step ahead. But today I'm not going to talk about it.
And
But this
Suparna,
works.
I
Yes?
need to encourage the audience. Don't forget to ask questions in the Mentimeter tool below the webinar stream. Thank you.
Yeah, thank you. So when we started culturing in bioreactor, we already see that it has incredible potential to go to very, very large scale. I mean, I have grown ribosomes previously commercially in the 100-liter scale. We realized that J28 can be grown in the same way in the bioreactor. But then when we are academician researchers. So when we get such a big volume of cells, what do we do that? How do we harvest it? So we still used batch centrifugation, but then as Patricia says, it's highly possible to use different kind of filtration technique to harvest cells. We haven't used it, but this is highly possible, I understand. Then the cells are lysed, and here the next step is the clearing the lysate, which is extremely important. So in this step, we first use this depth filtration using a Pall filter, and I will come to that. So before doing that, I say that, earlier we used to clear the lysate by 2 times centrifugation, low speed centrifugation, like 20,000 RPM. So let's say 20,000 RPM 2 times. And despite that, it could have been possible that it was not fully cleared. So we needed to pass through like lab scale, very small filters in order to clear it up. But when we did it in the Testa Center in collaboration with Testa and Pall Technologies, Patricia was particularly to help, that we could choose a Pall PDH4 filter that you see here. So this is a depth filtration media. I stole this cartoon just from Patricia's talk a few minutes before. So it really shows that how the contaminants are stopped actually largely in the depth filter matrix. So what you see here in the left, it's a very simple peristaltic pump. It goes through a pressure gauge, and this is a pressure meter. And then the sample is going to the filter, and it is being filtered here. The debris will be accumulated here in the depth filter, and then the cleared lysate will be collected. And we were surprised how efficient and effective, not just efficient, how effective this was. So definitely, what are the advantages? The advantages is that it's fast and easy. One can filter as big volume as possible. One is not limited by the size of the rotor. Secondly, that my group members clearly tell that they realize that it's much better separation of cell debris from the cleared lysate compared to centrifugation method. And third, very important for us because ribosomes are so precious, we see that in this method there is less loss of sample. Otherwise, when we centrifuge, a lot of ribosomes actually go to the cell debris, which we cannot retrieve. And that was not the case in this filter. If you ask me why, I cannot answer at the point. We need to investigate more, but that is the situation. We ran this, they ran this at, you can say, 10 ml per minute. But if you choose, like Patricia said, much bigger filters, then the pump speed can be made much higher. So that means this is a very effective and efficient step and can be used for clearing lysate, not only for ribosomes, but for many other things. And we have experienced it. So after that, the cleared lysate we put in the affinity chromatography using one Acta Pure system. And here you can see that we packed a 200 ml column. That was enough for the scale we worked with, like roughly 40 grams of cells. And we used a pretty high flow rate, 15 to 20 ml per minute, and ran imidazole gradient. Here, what is in the screen, you can see here. This is the elution profile. And for the UV detector, we used 250 nanometer wavelength. The reason is, I told, ribosome is made up of mainly RNA. So we followed the elution based on the nucleic acid absorbance. So here you can see that this is non-specific binders which are being washed out. And then here comes the peak. And since each ribosome has 4 his columns, it's pretty tight binder to the column. And this sharp peak is the ribosome peak. These are the eluted ribosomes. So then what to do with the eluted ribosomes? So one possibility was that which we tried to compare. Again, put them in ultracentrifuge and spin them down to concentrate. But then we used actually for concentrating ribosome a diafiltration technology in the Actaflux system. And we used this Pall Minimet 300 kilodalton filter. I already told you that the ribosomes are 2.5 megadalton. So definitely they will be concentrated through this filter. So we used one Actaflux system in the Testa Center. And I briefly explained this. So here is the main important thing. This is a sample container. And you can see that it has very well the volume marked on it. And its weight is being monitored. So we put the sample here. And the same system one can use for buffer exchange, like what Patricia mentioned. And here is your buffer container. So this is the inlet for the buffer. And then the sample is going through this tubing through the pump. And then this is the inlet, and it's going to this Pall Minimet filter. And then here, through the filter, you can see that these yellow valves, these are actually outlet valves. So when it is getting concentrated, the sample, the concentrated ribosome is actually coming back through this channel in the sample container. And then this pathway continues, like iterates. And only thing that is actually the cutoff which they set is the weight of this sample container. So when the weight becomes low, it will pump in more buffer or take in more sample, and then it will concentrate it. And the excess buffer, which is exactly what happens, the filtration in our kidney, in our glomerulus, in the same way that the aqueous media is actually going out through this outlet and being collected here.
So
this is a, yes?
In the interest of time, I need you to wrap up your presentation in like a minute or two. I hope.
Yeah, it's one more slide. No problem. So basically, this is the great advantage that the same filters one can use to concentrate and also to change buffer. And then you can see that, yeah, we purified the ribosomes. These are the typical ribosomal RNA bands on agarose gel. And then further, they checked the activity, and the activity was very high. The activity in synthesizing green fluorescent protein, and it was very high. So that means we managed actually to save a lot of time by clearing the lysate using depth filtration and then concentrating ribosomes by using diafiltration. So if I summarize, so then in conventional method, this I already mentioned that you need to centrifuge at 20,000 RPM and your volume is limited and you need minimum 1 hour. Whereas here, there is no limitation of the volume when you use filtration and you can use a fast rate of flow. And then more interesting is the concentrating the ribosomes, where we are dependent earlier only on ultracentrifugation on 30% sucrose solution, and then the total volume is again limited, and we had to do extra dialysis for buffer exchange, whereas in this one, all these things can be done with a filtration, where we use this Pall Minimet 300 kD filter, no sucrose needed, no limitation in volume, time took hardly half an hour, and no dialysis needed. So the added benefits, I would say that if we just look at this, we produced about 300,000 picomoles of ribosome versus 80,000 picomole in the conventional prep. Time investment was hardly 12 hours compared to 7 days. And the instruments were highly simplified, affinity columns and diafiltration compared to ultracentrifugation, and activity is higher. So I think this method can be applied just not to the ribosomes, but to other cell organelle or virus particle or other macromolecular complex. So with that, I will like to thank mainly these two group members, my senior researcher from my group, Chandrasekhar Mandava, who originally designed J28 ribosome purification and published in Nucleic Acids Research. And then Narayan Parajuli, a PhD student, they together developed this entire method. And thanks to Testa Center Arsenal, led by Jesper, and Charlotte was our direct contact point in Pall Biotech Patricia. And of course, we need to thank Uppsala University and our coordinators, Margareta Krabbe and Erik Jakobsen, for making this happen. I also extend my thanks to my funding agencies, key funding agencies, Swedish Research Council, Vetenskapsrådet, and Knut and Alice Wallenberg Foundation. Thank you for your attention.
Thank you, Suparna. Thank you. So we have questions from the audience. I think we have the time for one question, and then we'll come back to the questions after Jesper's presentation. And one direct question here. Now you're sharing your screen. If you don't want to do that, I suggest you stop sharing.
I managed.
No worry. Do you find that ribosomes aggregate upon isolation, purification, or concentration stages?
In this particular, I guess we are talking about what we did in Testa Center. We did not find any such problem by using this filtration technique.
Okay, so we'll come back to more questions for you after Jesper's presentation. Thank you, Suparna.
Thank you.
So Jesper, you are ready in the lab. Let's see where you are.
Am.
There you are.
Yes.
And for those who don't know you, you've been a student of the Uppsala University and a PhD at the Karolinska Institute. You've been with GE and then AstraZeneca for a couple of years, 6 years or so, and then GE again, totaling 13 years in GE, now Cytiva, as I understand it. I've seen you in this Testa Center since the beginning, and you've seen many projects come and go in the Testa Center. I think you will be perfect taking this presentation or this webinar away with the last presentation of the Testa Center. And something that you didn't know about Jesper, he has suddenly a lot of time in his hands, since he's usually oriented like competition where you run with a map. And if you have any questions on where you are, you should ask Jesper, because he's an expert finding you the way where to go. But this competition, the big competition in Sweden, which is called O-ringen, has been cancelled due to the pandemic and postponed until later next year, I think it is. Which Jesper, I know you're very involved in. So with that, please.
Thanks, Jens. Yes, I run orienteering, which is the Swedish way of trying to find your way through the woods with a map and compass. But that's another subject. But if you have interest in that, please contact me as well. So thanks a lot, Jens. I'll wrap up this webinar from a little bit more helicopter perspective, from a Testa Center perspective. And thanks a lot, Patricia and Suparna, for very insightful talks. I will try to communicate to the audience how we work here in Testa Center with the focus on the together, so that we together are able to drive projects through Testa Center, just like Suparna explained. So I'll share my slides, just like everyone else. I have slides, and you should see it now.
Yes.
Please, Jens.
And
yes.
And the questions are also open for Jesper in the bottom of the page.
Yes. So I will not take a lot of time. I'll show a couple of small slides around the instrumentation that we have here in Testa Center and what you can do in Testa Center. So we're a test bed for both biology and technology innovation around biological production. Suparna has just described a project around biological innovation, where she made a scalable process here in Testa Center. Our vision is that we together accelerate the journey of world-changing innovations. And this together is the direct intention of mine to focus a little bit on. Since we, together with the Paul and Patricia, together with the clients in Testa Center, and together with the experts here in Testa Center, we try to give the best service to clients I'm in a very active lab now, so I have a little bit of background noise. Please don't let that disturb you. But I try to find to broadcast from active labs. It's much more fun than sitting by a desk. So this is ongoing work here in one of our 4 bioprocessing labs.
Perfect.
Our mission is that we should enable researchers to verify innovation and to do that in a production environment, which means that it's relevant for scalable production. And by being in a relevant environment, we can also enable the researcher to accelerate innovation and industrialization of those inventions. And then we need to secure excellence. Biological production is a rapidly growing field, and we need to educate our students and also educate our research community in biological production. It's a joint effort between Cytiva and the Swedish government to support the growth of life science. Testa Center is a separate legal entity, but it's defined as non-profit. And we're open both internationally and nationally for academic groups and small companies around both biological and technological innovation. We describe ourselves as an industrial infrastructure lab, which means that we'll have the production equipment that is usually used in a GMP setting, but in a research environment here in Testa Center. So we're non-GMP, but you can run experiments up to 500-liter bioreactors and with the matching harvest and downstream scales for that. And then we're also open for educational activities. And Uppsala University, with Margareta Krabbe and Erik Jakobsson in the forefront, are driving several educational student courses here in Testa Center. So what can you get when you come to Testa Center? So you can get access to a lab, which I am in right now, which is a lab designed for bioprocesses. It's designed to harbor process from start to finish. Suparna showed you just such a process where you start with the cell bank, a frozen vial, you do the seed expansion, and you do the bioreactor cultivation. She did a harvest, and then she did a protein purification as well as a protein concentration in the end. So all the way from start down to purified protein, whatever protein that may be. Along the way, you will need perhaps some technical support, so we can provide that. Patricia is definitely a resource of ours. We consider you that, Patricia. So Patricia participates in many projects here in Testa Center, but also, of course, the specialists here in Testa Center will support the projects as they are performed there. We have 2 upstream specialists, so that is Adam Begelius and Erik Felt. And then we'll have a downstream specialist, which is Charlotte Brink. And together with Patricia, we have quite a good team that will support the projects being executed here in Testa Center. Instrumentation-wise, as we said, all the way from start to finish. So bioreactors in 1 to 5 liter scale, all the way up to 500 liter scale. We will, during May, commission a 200 liter bioreactor for microbial use as well. Harvest and filtration equipment. And since we're on the Cytiva site, we are a protein separation company by heritage. We have basically any piece of equipment that you might envision around protein purification. So all the way from the ECTA Start, which is the smallest, up to the full-fledged production system, ECTA Bioprocess. We also have some analytical capability to be able to run processes. And so in terms of cell counters, metabolite analyzers, HPLCs, and 4 Western blots.
So Jesper, we're running up on time due to some, we're running late a little bit, but I think we will continue since we still have a lot of viewers on the stream, and I would encourage the audience that if you have any questions or you have to run, please send us a message in the question box, and we will follow up any of your questions that you may have. Or if you're interested in being contacted by Testa Center, tell us that in the chat, in the question box, and we'll get back to you with information. So with that, please, Jesper, give us a couple of more minutes, and let's aim for questions in like 5 minutes or so.
Yes, I'll show 2 more slides actually. One slide which might be interesting for many. If you have a project where you're aiming for a commercial endpoint at some point, you can apply for funding from Vinnova. So please contact us if you are interested in that. There's always a need for funding, I'm sure, when you try to drive a commercial process. And I'll end very shortly with the Testa Center in 30 seconds. So we are non-profit, we provide expert support, and we're a non-GMP lab for biological production. And we're a private-public partnership between the Swedish government and Cytiva. And when you come here, you get access to production equipment, a unique equipment park. You get access to our expertise, and you pay per usage only, so that you don't need necessarily to do the capex for a 500-liter bioreactor, and you get a professional network. Many of the projects achieve a lot of things when they come here, or most of the projects, but the common thing that they almost always look for is to reduce risk when going up in scale. So going up in scale is accompanied with some technical risks, and being able to do that in an environment which is non-GMP, and when you get the expert support to sit side by side when you do the experiments, is very beneficial for many other projects. So reducing risk and also increasing the know-how of your own process. And together those two increases the value of your project significantly. We have several examples of that. Very important to say, when you are in Testa Center, you generate results and potential IP. Those results and IP is yours. So Testa Center will not claim IP, and if you want to do your project under confidentiality, it's perfectly fine for us to sign a CDA or an NDA. With that, I think I will wrap up actually, and just say that if you have any questions that you would like to shoot over to me by email, please do so. We're here to support you. Thank you, and over to you, Jens, I guess. And if we have any questions, I'll be happy to answer them.
Thank you, Jesper. Thank you. So I will check the questions, and you can still ask us questions in the question box. And then there is like 1 minute feedback question also. So let's see. I'll just quickly jump into the questions and just shoot to whoever it's assigned to. All right. So let's see. So one question for Suparna. What was the main benefit from utilizing Testa Center for you and your project?
The main benefit, scaling up and doing in a very time effective way.
Nice, short and sweet. Very good. And we have some really technical questions for Patricia also. Let's see. Okay, so what would be recommended for COVID-19 PPE to apply for bioprocess applications and manufacturing facilities?
So if I understood correctly, the question, it is what kind of PPE one would have to use for an environment, for a work environment? Was that it?
So PPE is personal protection?
Yes, exactly. Well, I can't put a claim on that because it's not my type of specialty, to be honest. I would say that you would need to follow local recommendations, and that really, if your workplace is responsible to check that and to provide appropriate protection. But then if it's something more, if you need to protect your product and your buffers, then I know a little bit more about that.
Is Jesper trying to get into the conversation?
No, I'm just stepping into the camera to be in picture.
All right, so we have a question for Professor Sanyal. Is all this protocol that you develop fully automated in a plug-and-play approach, or is it something that you have to follow your instinct and empirical evidence?
We are going towards fully automated protocol. Most of the steps, it's so standardized that you do not need intuition.
Okay, so we have a couple of more questions and they are pretty technical, and some of them would like to be contacted by us in person. So with that, I think we will close this webinar, and we will get back to you who still have questions open individually afterwards by email. So with that, I would like to thank all the presenters. Thank you very much for participating. I hope to see you again. And to the audience, thank you very much for participating. And there is a question now, which is, would you like us to get in contact with you? Please add your email or phone number or something similar if that's the case. And by that, I wish you a good day. Have a great lunch to everyone in Sweden if you not already had it. And take care. Bye-bye.
Thank you. Bye-bye.
All right. Thank you very much.
It was lovely to join.
Lovely. Bye-bye.
Bye-bye.